How did ancient TB reach the Americas, and what were the dynamics of this disease over time?
We really want to focus upon the lives of the people whom we have the privilege of studying. In order to do that, we have to engage in interdisciplinary study. So we need to bring in the paleo diet, the paleomobility people, so we know how people moved across the landscape. And we also need in-depth information from the archaeological record to learn more about diet, about the animals with whom our people interacted, and reconstruct lifeways as best we can.
In our research project, Osmore Mobility and Infectious Disease, we're asking the question, how did tuberculosis spread within and among communities living in the Osmore Valley of southern Peru? To answer this question, we're using archaeological and osteological data, ancient DNA data, and stable isotope data. Between about 400 to 1500 CE, skeletal evidence from southern Peru and northern Chile shows that many individuals suffered from TB. Tuberculosis, or TB, is a bacterial infection caused by Mycobacterium tuberculosis and its close relatives. TB is still a major cause of deaths worldwide. It commonly affects the lungs and causes symptoms like persistent coughing and chest pain. And in some cases, TB disseminates to the bones. In these cases, where it causes distinctive lesions, particularly in the vertebra of the lower back, it can be identified in the archaeological record. Before European contact, ancient TB cases are found throughout the Americas, but primarily in densely populated regions. The first cases appear in the archaeological record in South America, and later in North America. Between 700 to 1300 CE, molecular and skeletal evidence from southern Peru and northern Chile shows that many individuals suffered from tuberculosis. Our previous research indicates that the populations of coastal regions in Chile and Peru, as well as inland Colombia, were affected by tuberculosis linked to Mycobacterium pinnipedi. Here you can see a family tree or phylogeny showing the relationships among the different TB strains. Mycobacterium pinnipedi is responsible for the spread of tuberculosis in seals and sea lions. This pathogen likely jumped from seals to humans through the butchering process or ingestion of undercooked meat. Evidence of seal hunting is found throughout the archaeological record in South America. This includes seal bones and depictions of seal hunting on moche pottery. Molecular studies also suggest that instead of one jump from animals to humans, there may have actually been several animal-to-human zoonotic spillovers. By analyzing the genetic diversity of ancient TB strains, we may be able to determine if there were indeed multiple zoonotic jumps that led to TB spreading through this region. We can also address the second goal of understanding the role of human mobility in spreading disease by using archaeological and isotope data to infer how connected different communities were to other communities. How do we use archaeological and paleopathological data to make inferences about disease in the past?
The first line of evidence we have for tuberculosis in the archaeological record comes from disease-altered bone from people in the past. We look at their spinal columns particularly because there is where the diagnostic, the pathognomic symptoms, the cavitaceous lesions occur.
How do we use ancient DNA data to make inferences about disease in the past? In the laboratory, we extract the genetic material of the disease-causing pathogens from bone samples taken from characteristic TB lesions. This DNA is then analyzed to understand its unique genetic makeup. By comparing it to the DNA from ancient and modern strains from around the world, we can understand the spread of this pathogen over time and potentially also how the strain adapted to humans. How do we study the stable isotopes and what do they tell us?
We use stable isotope analysis in archaeology to understand where people were living in the past. And we can do that because Isotopes get incorporated from the environment, where they vary based on either the geology of the region, in the case of strontium, or the larger environment, in the case of oxygen. And those isotopes get incorporated into our bodies through the food we eat and the water that we drink. And because different parts of our bodies are growing and forming at different times, that means that we can look at isotopes in different bones and in different teeth to look at where someone was living at different times in their lives. And in this project, we'll use those data to understand how people's mobility is being impacted by or is related to tuberculosis in the region.
We also thank the people of the past for allowing us to share their lives, and the people of Peru who have let us access the important remains there that have fueled these analyses.